Hello students, a very good afternoon to all of you. So I'm Vinay. So today we'll be talking about molecular basis of inheritance. Okay. So before we have an introduction to this chapter, so let us see what you understand. So let me check. Okay, students, first of all, I will launch a poll on technical check. Please let me know if my voice, that is audio on the video is clear. Okay. The students from the YouTube, you can just put it in the chat box. Okay, so that it will help me. Okay. All right, students, you can just let me know if you have any problems with the session with respect to audio or video, so we can correct it during the course of a session. Okay. So first, before we start the basics and introduction to molecular basis of inheritance, so let us solve some question. So we have a first question, that is genetic material, DNA of a cell is found inside. Okay. So we have the following options, that is ribosomes, okay, lysosomes, nucleus, and endoplasmic reticulum. So I launch an MCQ poll. Okay, you can try to answer. It's all right if you go wrong. Okay. You have 1 minute to solve this question. So try to answer as quickly as possible. Okay, your time is up. Okay, so we are almost reaching 50 seconds. And it's a very simple question, so I'll end the poll right now. So the question asks, where is the genetic material, that is the DNA, found inside our cells? So the answer, the very logical answer for that would be nucleus, because we know that even though we don't know, if you don't know that DNA does not exist in nucleus, but still you know something, that the nucleus controls the entire activities of the cell, right? So the nucleus gets this capacity to control the entire cell because of the presence of the genetic material, that is DNA, within the nucleus. So when it comes to ribosomes, lysosomes, and endoplasmic reticulum, they do not show any genetic material. So these are the wrong options. So right option is option 3. So we have another question. Okay. So in another question, what do we have? We have genetic information is carried out by long chain molecule made up of amino acids, enzymes, nucleic acids, and histone proteins. Okay. So these are your options. Let me launch a poll again. Okay. Let me launch a poll again, and you try to answer within 1 minute. Okay. Yeah. So the poll is launched. You have 1 minute to answer this question. Okay, 30 seconds up, students. So try to answer it as soon as possible. Okay, so let's end the poll because it's a very simple question. If you have studied biomolecules in your first year, so it would be really, really helpful for you. Yet, so the answer for this question would be nucleotides. Okay, so why nucleotides? Because our DNA is made up of, okay, our DNA is called as a heteropolymer of nucleotides. So our DNA consists of nucleotides like A, T, C, G, okay, which are arranged in different fashions and they form this long chain, okay. So the answer is nucleotides. Whereas when it comes to amino acids are the monomers of proteins, and enzymes are, biocatalysts, and they're also proteins, especially. So when it comes to histones, so we'll study about histones and how the charge of the histone is very helpful for the packing of our DNA. Okay. So for the question, the answer is nucleotides. So let us get back to the introduction to molecular biology. So molecular biology is a science where we study, okay, the genetic the molecular basis of the characteristics. Okay, so if you have any expression, any character, or if you talk about paleontology, or if you talk about evolutionary relationships, or with respect to family history, or any of the aspects, if you want to know to the core, the complete ground level of it, molecular basis is the key for it. Why? Because we always question something about, are we related? Okay, are we different from each other when it comes to living system? But for the answer for all this question lies with understanding molecular sciences. So molecular sciences gives us an idea that all living organisms, okay, most of all the living organisms, from bacteria to the most complex eukaryotes, all of us have the same genetic material, that is DNA. Okay, DNA as our genetic material, and the composition of our genetic material is also the same. The same nucleotides are present in plants, and the same nucleotides are present in animals, and even in the microorganisms. So as we study more and more about molecular biology, we understand the scope of this particular subject. It not only allows us to understand the very basis of life, how the life gets its character, or how an organism has this different diversities, etc., but also it gives us a tremendous application for human society. For example, it might be agriculture, it might be forensics, it might be medical sciences, okay, it might be agriculture. You take any of these cases, the application of molecular biology is the cutting edge technology right now. Okay, but it did not start as the huge boom as what we see now. Okay, molecular biology has evolved a lot compared to the last decades. Okay, last few decades. In within 2 decades, like in 20 years, the molecular sciences have completely changed the perspectives of human approach to biology. Okay, so if you look back in the past and see, did it happen overnight, like how molecular biology is influencing human society or our lives? So it did not start like that. And it started with the basic question of man to understand who carries the characteristics from one generation to another generation, or are their characteristics transferred from one generation to another generation. So we have come across several prehistoric theories, and we have understood, okay, why they did not succeed in explaining about the genetics of inheritance, right? So until a brilliant man, that is Gregor Mendel, when he worked on his pea plants, and he tried to propose that there is a possibility of existence of factors, okay, and the factors behave in a particular fashion in case of dominance or segregation or law of independent assortments. Okay, so Mendel was trying to give his idea through his experimental proof that inheritance happens, and there is something transferred between the parents and the offsprings, right? So after Mendel worked tediously with his genetics experiment, okay, but he could not prove that what is being transferred from one generation to another generation. Mendel, throughout his experimentation, he talks about factors, but he never talks about genes or DNA as being the specific molecular, molecular thing that is passing from one generation to another generation. So after Gregor's Mendel work, it took almost 90 years, okay, for the rest of the proofs to be rediscovered or discovered, to give more substantial evidence to molecular inheritance, okay. So what happened, it all began with simple experiments, okay. For example, in 1977, Oswald Avery, what he did, he proved that, okay, let me change the ink. Okay, Oswald Avery, what he does is he proves that DNA is a genetic material, okay, that is responsible for DNA is a genetic material, he says. So when he says that DNA is a genetic material that carries the information, it was not just Oswald's idea. So before Oswald, there was another scientist called as Griffith, who would have performed on transformation experiments. We will talk about it. So Griffith's experiment gave an idea to Oswald to just try to prove what is the molecule that is responsible for the genetic material. So he proves that DNA is a genetic material. And after the Cheggs comes, and he tells about the ratio of the nucleotides that is present in a DNA. So DNA is a very structurally stable molecule. So when you talk about DNA structure, he also talks about a mathematical ratio that is present in the DNA, okay. So here we understood that what is the ratio of purines to pyrimidines, okay, we'll talk more about it. Next comes your Watson and Crick. So so next one is Franklin, okay. Franklin and Wilkins, what they do is they perform an X-ray diffraction studies on DNA. So they take the X-ray images of DNA, okay, by crystallography. After they conduct this X-ray crystallography, they would not be able to give a proper statement, okay, or a proper statements on what is the structure of the DNA. So they will fail to explain it. So using this X-ray crystallography studies in 1953, Watson and Crick, okay, they propose something, a revolutionary double helix structure of DNA. So understanding the structure of DNA really opened up a different world, where we started understanding too many things about the characteristic feature of DNA, okay. By 1961, okay, we understood that DNA carries information and they are expressed in the form of codes, okay. So the genetic codes were the concept of genetic codes were understood by 1961. Okay, by 1970, Cohen, they developed recombinant DNA technology. Here is the biggest step, where from your discoveries, we just step into another world called as biotechnology and molecular biology. So with the discovery of recombinant DNA technology, okay, next we have DNA sequencing, okay. Sanger, we understood how to decode our DNA, okay, how to open up our DNA and decode the codes that are present in our DNA and try to store that information, okay. So next, what happens in 1983, okay, in 1983, the PCR, that is polymerase chain reaction, or synthesis of artificial DNA in higher quantities was understood by 1983. In 1984, fingerprinting, okay, this is another revolutionary innovation, I can tell. So fingerprinting really changed the world in understanding our history, okay, and criminology and forensics, okay, and in any of the cases, it gives accurate evidences on who this person is, or what this organism is, and who are the close relatives, etc. So in 2002, after that, we have the first companies to commercially synthesize genes. So we started using genetic engineering tools to really synthesize certain enzymes and proteins, etc. That is where the industrialization began with respect to biology, right? So by the year 2002 to 2003, okay, one of the mega projects was conducted. Okay, it started in 1993. Okay, by 2003 it was completed. So this project is called as Human Genome Project, which is one of the mega projects in the entire biological sciences. Okay, so by 2008, okay, genoscripts, like what do you tell? We started developing synthetic genes, synthesis of genes. Okay, so these gene scripts were developed by 2008. Okay, so next by 2012, okay, uh, they joined the project where they start building this eukaryotic genome using yeast. Okay, so they start developing a synthetic gene script for eukaryotic genome, that is with the yeast. By 2013, okay, so the discoveries that were made in 1953, okay, and the published whatever the letter the Crick used to publish the double helix, so it was sold for astounding $5.3 million in an auction, okay, in 2013. So let us get back to, okay, let us get back to the basics of molecular biology. So what happens is, before we talk about the specifics of this subject, I told you it all starts with the discovery of an important thing called as nuclein. So Frederick Michener identified nuclein. Okay, he identified DNA and he named it as nuclein. So next, what happens after a few years, James Watson and Francis Crick, they proposed this model of DNA based on the X-ray diffraction. So this X-ray diffraction was conducted by Wilkins and Franklin. So before we understand how the DNA was discovered and how did we come into the how do we come to the conclusion that DNA is a genetic material? So let us go back to the Watson and Crick model of DNA. Okay, so first, what happened once the nuclein was discovered, X-ray diffraction studies were conducted, and using this X-ray diffraction studies, Watson and Crick proposed this double helical structure of DNA. So in this structure, what he says is the DNA is made up of two polynucleotide chains. Okay. So when we say that it is made up of two polynucleotide chain, that means there are two chains, right? There are two polymer chains that are present in the DNA, and they are coiled in a helical fashion. So helical fashion is like a staircase. So whenever we draw a DNA, so we draw it like this, right? You can see in the picture also. So that means what our DNA is coiled like this. Okay. So it is coiled in what direction has it coiled? It is coiled in a right-handed fashion. So the DNA that we study from the Watson's model is called as bDNA. Okay, we call it as bDNA, or we call it as right-handed helical DNA also. Okay, so here, when we observe the structure of our bDNA, so basically we see these turns that is happening sequentially, right? So you can see that here is one turn and here is another turn. So for every turn, okay, that happens between every turn, right? So one turn to another turn, this region is called as a pitch. Okay, this region is called as a pitch. So a pitch is usually around 3.4 nanometers. So from one turn to another turn is called as a pitch, and each pitch is around 3.4 nanometers in length, or it is 34 degree angstroms. So in one pitch, okay, it contains around 10 nucleotides. So for every turn that it takes, okay, from one turn to another turn, that the pitch contains around 10 base pairs, right? So the distance between each base pairs would be naturally 0.34 nanometers or 3.4 degree angstroms. All right. So this is a very basic thing that we have to understand. So when we try to analyze the structure of the DNA, so when we see the DNA, the diameter of the DNA is around 20 degree angstroms. Okay, so it's around 20 degree angstroms. So both the strands that we have, right, we have two polynucleotide chains, and both these polynucleotide chains that are there, they are held together by hydrogen bonds. Okay, they're held together by hydrogen bonds, and these hydrogen bonds are between the nucleotides, okay, or nitrogenous base pairs that are present in individual strands. So what happens is, if you have some of the nucleotides in one strand, and you have the other nucleotides in the other strand. So one of the important characteristic feature of the double helical DNA or DNA is its complementary nature. So what is complementary nature? If you have adenine in one strand, so adenine always binds to thymine, okay? It always goes and attaches to thymine using two hydrogen bonds. Whereas you have cytosine, which always binds with guanine with three hydrogen bond. So this complementation is called as base complementation. So through base complementation, what happens? The bases that are in one strand is attaches to the other strand using hydrogen bonds, two hydrogen bonds or three hydrogen bonds, okay, based on whether it is AT or CG. So now if you see the entire DNA structure, our entire DNA structure is made up of nucleotides. And if you understand what is nucleotides, nucleotides are basically made up of a pentose sugar. You can see in this diagram. Okay, so they basically contain this pentose sugar. And second one they have is a nitrogenous base. Okay, and then they have is a phosphate group, right? So this is what is a nucleotide. So when they make up the constituent of a DNA, so if you observe the DNA is made up of two strands, right? So in these two strands, what you can see is you can just imagine a structure like a ladder, okay, and you can just imagine the sticks that are in the ladder. So whatever this frame of this ladder, okay, this portion and this portion of this ladder that we are talking about, let us call this as a backbone. So our DNA also resembles to that of a ladder. You can see here, right? So let us call this as a backbone of this DNA. So the backbone of this DNA contains two important ingredients or components. That is your sugar, which is your deoxyribose sugar plus a phosphate. Okay, a phosphate group. They make up the backbone of your DNA. Okay, whereas your nitrogenous bases that we were talking about, okay, which makes the hydrogen bonds, they try to project towards the inside. Not try, they project towards the inside. Okay, you can see here you have your pentose sugar and then you have your phosphate group which make up your DNA, right? The backbone of your DNA. Whereas your nitrogenous base is what they do, they project towards inside. When they project towards inside, it becomes very easy for them to bind with other nitrogenous base pairs. Okay, so here when we talk about nitrogenous base pairs or nucleotides, when we talk about nucleotides can be of two types, right? So one we call it as purines, other one we call it as pyrimidines. So what do we call as purines? Purines are, okay, see, for example, if I tell purine, right? Purine's example is A and G, adenine and guanine. Okay, so why do we call adenine and guanine as purines? Is because they are made up of, if you see the nitrogenous base structure, so they contain two rings. Okay, they contain two rings in their structure. Okay, so something like this. Okay, their nitrogen base structure contains two rings. So one ring is a 6-atom ring and other ring is a 5-atom ring. So the 6-atom ring we call it as pyrimidine. Okay, pyrimidine. Okay, sorry, pyrimidine ring, and other one is called as imidazole. Okay, imidazole. And why do we call it? See, 5 ring is called as imidazole, and why do we call it as imidazole is because it contains around 3 carbon and 2 nitrogen. And why do we call it as pyrimidine is because it contains 4 carbon and 2 nitrogen. Okay, so since it contains two different rings, they are also called as heterocyclic compounds. Which one? Purines. Okay, so when it comes to pyrimidines, for example is, okay, for pyrimidines the example is cytosine, thymine and uracil. Okay, they contain only one ring, that is the pyrimidine ring. Okay, so that is why they are called as pyrimidines. So usually what happens, always a purine comes opposite to a pyrimidine. Okay, whenever the purine comes opposite to a pyrimidine, it will uniformly create this stacked structure. Like if you arrange the coins, 1 rupee coins, one upon each other equidistantly, right? So you can see that there is a very uniform arrangement, right? So that is why your DNA will not go up and down. It is very uniformly stacked. Why? Always a purine. For example, adenine is a purine, thymine is a pyrimidine, correct? Cytosine is a pyrimidine, guanine is a purine, right? They come opposite to each other. That will give the DNA a stacked structure. So when he was proposing the structure of DNA, they also talk about anti-parallel nature of DNA. So what is anti-parallel nature of DNA? I'll just try to explain here. So if you see the DNA structure, right? Yeah, when you see the DNA structure, the DNA has two strands, right? Hold on. Yeah, I'll just change the ink. Yeah. One second. I'll just change the ink to white. Yeah. So we have two strands in the DNA. So one strand is called as 5 prime to 3 prime, and the other strand goes from 3 prime to 5 prime. Now, what is this 5 prime and 3 prime? So 5 prime and 3 prime, they're actually denote the polarity or the direction of the strand. So how do we recognize which strand is 5 prime and which strand is 3 prime? So if I take the tip of this strand, okay, if I take the end of this one strand, so in this one strand, what did I tell? Our DNA is made up of, okay, a sugar group, that is a pentose sugar, that is deoxyribose, and then they have a phosphate group here, correct? No, and here this in third carbon atom, what happens? There is an OH group, okay, and here what do we have? We have a nitrogen base, correct? No, so here what happens is, if I see the tip of this one DNA where I have written 5 prime, I will see a free phosphate group, okay, there will be a free phosphate group. Whereas if I see that tip of the same strand, okay, on the opposite side, what do I see is there is a free OH group. Here there will be free OH, here there will be free phosphate. Same vice versa. The other strand will be opposite. If it is phosphate, here it will be OH here. Okay, here it will be phosphate. So this is called as anti-parallel nature. Okay, anti-parallel nature of DNA. So they talk about base complementation, and they also talk about how the strands are anti-parallel to each other. Correct? No. So this is about the structure of DNA. So when it comes to DNA and RNA, both are made up of nucleotides. But DNA is very structurally stable molecule, like we can call it as, whereas RNA is not so stable. It is highly reactive. Now, what is the difference between DNA and RNA structures? So DNA, they have only one OH group at the third position. Okay, let me change the ink again. So yeah, so see here in the third position there is only one OH, right? But if you compare with your RNA structure, you can see that both your third carbon, this is third carbon and this is the second carbon of your sugar. See, whatever we have written, this is a sugar, right? So here if you see in the second carbon atom also there is a OH group, and in the third carbon atom also there is a OH group. Because of this extra OH group that is present in the second position of your RNA, it becomes exceptionally unstable. All right, and moreover, RNA is single stranded. It is, if it is double stranded, RNA is pretty useless. Okay, so DNA is double stranded and it is a very stable structure. So that is first difference. DNA is highly structurally stable. RNA is not stable. It is catalytic. It is highly reactive. Okay, why is the reactivity? Because of the presence of extra OH group. Now, if you see the type of nucleotides that is present in DNA and RNA, in DNA, you have your nitrogen bases, that is adenine, thymine, cytosine, and guanine. Whereas you have the nucleotides in RNA as adenine, uracil, cytosine, and guanine, AUCG. So you can see here, instead of thymine, the RNA has uracil. Okay. So because of this additional stability, they're structurally and chemically stable. The DNA is structurally and chemically stable, and it involves in so many different aspects of our molecular sciences, which we'll be studying. So that is why DNA is genetic material in most of the organisms. Okay, so as the structure of the DNA was proposed, before the structure of the DNA was proposed, Chergov had proposed his rule called as Chergov's rule, where in his rule he tries to give us an equation to the purine to pyrimidine ratio. So for what happens is, he says that if you have, okay, adenine and guanine, which is a purine, thymine and cytosine are pyrimidines, right? So if you add adenine, the number of adenine plus the number of guanine. So the total sum should be equal to the total number of cytosine and thymine that is present in our DNA structure. Or we can also put this equation as A plus G divided by T plus C is equals to 1. Always in a DNA structure, this ratio is constant. But sometimes what happens is, in some DNAs, the Cheggs rule might shift. Okay, so based on the purine to pyrimidine ratio, it might quite change. Okay, so it is not always that it will remain 1. There are many cases, okay, where your ratio will is either below 1 or above 1. Okay, so this is about Cheggs rule. Now, using this Cheggs rule, let us solve the question. So you try to do this question. You have the equation already. So let us see the question first. In a double-stranded DNA, okay, 1 second. In a double-stranded DNA, it has 20% of thymine. Calculate the percent of adenine in the DNA. So that is your question, and here are the options: 40%, 30%, 20%, and 10% are the option. So I will launch a poll and I will give you 1 minute to solve this question. Okay, and you can answer it using the polls. Okay. YouTube students, if you have any problem, you can just put it in the chat box if you have any doubts. Okay. And you can also answer in your chat box. Okay, where are we? How many minutes? Yes, we have 5 seconds left. Did you get your answer? So what's your answer? Okay, fine. If you have your right answer, it's good. If you have your wrong answer, it doesn't matter. All right. But try to answer, because it will be very easy for us to communicate and understand if you're like really understanding the class or not. So don't be ideal audience. So be an interactive audience. It will be very nice. Okay. So they've told that it is 20% of thymine, right? So if we take 100% of the DNA, out of which you have 20% of your thymine. So what is our formula, people? Our formula is adenine plus guanine is equals to cytosine plus thymine, isn't it? Correct, no? So there is a trick here. Okay, there is a very small trick here. If thymine is 20%, thymine always goes with adenine, isn't it? So if thymine is 20%, then adenine should be how many percent, people? This is the first trick. Okay, so adenine will also be 20%. Correct, no? So if adenine is 20% and thymine is 20%, the total amount is 40%. So out of 100% of the DNA, we have 40% of A and T. What is the rest? Rest is 60%. So out of 60%, again, we have to do it half. That means what? If guanine is 30%, obviously you have cytosine will be how much? 30%. Simple, isn't it? So this is one way of solving it. And the second way, we can go in a standardized way. How do we do that? So it has to equate to 1, correct? No, it has to equate to 1. That is A plus G divided by C plus T is equals to 1. You can take it as, so adenine is equals to thymine, right? So we have 20 plus G divided by, C plus 20. So finally we get, how many percent we get? Yeah, they have asked about thymine, isn't it? So thymine will be obviously 20%. If thymine is 20%, we will get adenine is also 20%, as simple as that. So our option is 20%. Okay, so you can use that ratio also to solve the question, or if you go with base complementation also, it will be easy for you to get the answer. Okay, so let us go back to a few more characteristic feature of the DNA. So what is DNA and how big is DNA? So when you talk about DNA is present in all living organisms, as I said. So in some of the cases, like viruses, they have RNA. So okay, some of the viruses have RNA, and they do not participate as genetic material. Okay, so correct this. So they do not participate as genetic material, even for their replication. I mean, like even if when they want to proliferate, they go convert their RNA into DNA and then start expressing. So DNA is universal when it comes to living organisms. So if you see the DNA, it is a very large molecule. Okay. So even if you take a simple organism like a bacteriophage, so bacteriophage, which is a virus, so it has around, okay, 5,386 nucleotides, okay, which is quite a large number for a virus, which is of so small size, right? So similarly, if you take an example, okay, 1 second. Yeah. If you take an example of other bacteriophages, okay, lambdaphages, so they have around 48,502 base pairs in the DNA structure. So E. coli, if you take 4.6 million base pairs they have in their overall DNA, okay, in their overall constituent of DNA, we have how many base pairs? That is 4.6 into 10 power 6, or 4.6 million base pairs are there. Come to human DNA. Human DNA is exaggerating 3.3 billion base pairs. Okay. So if you just keep unwinding your genetic material, you can go from Earth to moon and moon to back. Okay. That would be the length of the DNA. And if I have to give you a fact, a human body is made up of 30 trillion cells on an average, 30 trillion. Okay. And all our living cells have DNA of the same volume. Okay. 3.3 billion base pairs. Now imagine the amount of information that is in our body and in our cells, right? So let us have an interesting topic to calculate. Okay, so let us calculate the length of the DNA. Now, what do we know about the number of base pairs in our DNA? So it is 3.3 into 10 power 9 base pairs, isn't it? So total number of bases would be 6.6 billion bases, right? So you just multiply 6.6 billion bases into the distance of bases. So we know that between each base, what is the distance? We have 0.34 nanometer, isn't it? So we take 0.34 nanometer and multiply with it. So our each cell has a DNA which is as long as 2.2 meters in length. Okay. Now imagine the structure of your cell and then try to correlate with the length of the DNA. So it might confuse you, like how this big structure is even present in your DNA, right? So before we go into where the DNA is present and how it is packed, right? So let us solve one more question. What is the nature of the strands of the DNA duplex? Okay. So you have 4 options: anti-parallel and complementary, identical and complementary, anti-parallel and non-complementary, non-identical and complementary. So I will launch a poll. So try to answer this question. Okay. Sorry, I didn't end the poll. Okay. Yeah. Here you have one more poll. You can try to answer students. Okay. What is the nature of the strands of the DNA duplex? So I have so YouTube students, I know you cannot see the poll, right? So you can try to answer in the chat box. Okay. So it will help us to understand better. The question is about what is the nature of the DNA duplex. All right. Okay, 1 minute is up. I'll end the poll now. So if you have the right option, that's great. So let us see. What is the nature of the DNA? I told that the nature of the DNA is, first of all, we have to understand that DNA is made up of what? 2 chains, right? And when we talk about the 2 chains, what do we have here? In 2 chains, do they run in the same direction? No. So when when I wrote like this, okay, a structure back then, I told that one chain, if it goes from 5 prime to 3 prime, the other chain will go from 3 prime to 5 prime direction. So this is called as anti-parallel, right? So they're parallel, but they're going in opposite direction, right? So I also told you why we call it as anti-parallel, because one end of the DNA has a free phosphate group, and the other end, on the opposite side of the DNA, you have a free OH group. So it's not the same, isn't it? So it is quite opposite. So when it comes to the with respect to polarity of the strands, correct? No, when it comes to polarity of the strands, so we have anti-parallel nature of the DNA. So what is complementary? Complementary nature is we have nucleotides in each of the DNA strands. So if we have adenine here, then definitely in the opposite strand there will be thymine. If we have cytosine here, definitely there will be guanine in the opposite strand. So it is anti-parallel and complementary, right? So our option A is right. So I have, let's do one thing. Yeah. So our next topic is packing of DNA helix. So I told you that human DNA is so large that it would take 2.2 meters of DNA to be put inside a cell, correct? No? So why is packing even required? See, DNA is a very important genetic material, okay, correct, right? So if any of a single base pair or a fragment of a DNA, or either it is not distributed or if it is missing when the cell division happens. So when the cell division happens, what happens? Not only the components of the cells divide, but also the genetic material is divided between the parents and the daughter cells, right? So during this nuclear division or the distribution of genetic material, if the equal amount of DNA is not distributed well, then there will be a serious problem for the daughter cells, correct? No? Why? Because it cannot have the exact characteristics as their parents. So we'll understand what happens if you miss certain base pairs in your DNA. So that is why all living organisms try to pack their information, very vital information, that is DNA, inside their cells. Okay. So this packing also has certain what do you tell, complexities. For example, in prokaryotes, which are one of the most primitive cells, the packing is not so complex like that of eukaryotes. In prokaryotes, what happens? The DNA is very naked and it is scattered within the cytoplasm, first thing. And in all the cases, like in both prokaryotes and eukaryotes, DNA is negatively charged. So what happens? They have some proteins called as non-histone proteins. Okay, I will write here. Okay, let me change the ink, people. Give me a second. Yeah. So if I take non-histone, okay, non-histone proteins, okay, so they contain non-histone proteins which are positively charged. So DNA is negatively charged. Now what happens? They are randomly coiled. It's not in uniform coiling. Okay, they're randomly coiled. So the positive and the negative charges attract each other, and then they will coil up as tight as possible. Or for example, if you have more positively charged protein groups, then there will be more amount of DNA that is being coiled. In other places, if you have less amount of proteins that are scattered, then the less amount of base pairs are packed within the same structure. So that is why the packing is not so uniform. And the packed DNA, usually the DNA that is present in your bacteria, it is called as nucleoid because of this type of packing that is present. Okay, so this is about prokaryotic packing. So when it comes to eukaryotic packing is highly systematic and it involves very powerful proteins called as histones, which are positively charged. One histone unit, okay, if I tell one histone unit is made up of 8 monomers of histones. Okay, it contains 8 histone monomers. So 8 histone monomers, they combine together to form this unit called as one histone octamer. Okay, they form one histone octamer. So one histone octamer, it can pack up to around 200 nucleotides around them. Okay, we'll talk about it. So what do these histones contain? Histones contain very highly rich amount of lysine and arginine in their amino acids. See, histones are proteins and proteins are made up of amino acids, right? So the nature of the amino acids determine the nature of the protein. It's as simple as that. So when you talk about lysine and arginine are the best examples for positively charged protein. So if the amount of lysine and arginine is very, very high in a protein structure, then obviously they will become positively charged, right? So these positively charged histone proteins, they combine together in the units of 8 to form histone octamer. So histone octamer, which is positively charged, it starts winding the DNA around it. Okay, it starts winding the DNA around it. And how much of the base pairs they can wind up? Around 200 base pairs they can wind. Whereas there is another histone, okay, which is called as H1 histone. So for example, if I have one histone here and another histone octamer here and the other histone octamer here, so here what happens, 200 base pairs is packed. So from here to here, there is one stretch of DNA no, that connects another ball or another histone octamer. See, for example, take a long rope, okay, or thread, you wind it around here. Okay, then after it is complete, you just pull a little bit of you leave a little bit of rope and then start winding again with the next octamer, right? Similarly, so whatever this DNA that is not being wound, so it is called as linker DNA. Okay, we call it as linker DNA. So this linker DNA are held by H1 histones. Okay, so H1 histones hold this DNA that is between two histone octamers. So we understood about histones and how they pack. So if you see the organization of the histones, so histones, what happens, here is one histone octamer, here is another histone octamer, here is another histone octamer, and it goes on like this, right? So now what they do is they wind up like this. So if you see under your microscope, they look like some pearl string or beads on the string, right? If you remember a thread, and then if you write if you write beads on them, how do they resemble? Like a string, no? Like a pearl chain. Similarly, your DNA also appears like this. So this structure, we call it as a nucleosome. Okay, if you want to know what is an amount of nucleosome that is present in the human DNA, so then you just have to divide, right? 6.6 billion base pairs divided by 200 base pairs per histones. So how many we have? 3.3 into 10 power 7 beads you can find in your nucleosome. Okay, that is a length of your nucleosome. All right, students, like let us stop here and then talk about supercoiling and more packing of DNA in the next class. Okay, since it's our first session, so I will try to give you an NPS form. Please try to fill it up, because I didn't have a lot of students in Zoom class from the beginning, so it was a little strange to talk alone. So anyways, you try to fill this form up, for instance, now, and then we can continue in the next class. All right. And YouTube students, you can just try to put your queries or any problems that you have with regarding to class, okay, or the pace of the class. If you didn't understand any concepts, you can put it in the chat box, so we can take care of it. Okay. Okay. Strength. Anyways, thank you for your response, students. Okay, so we'll catch up in the next class, next session, and try to learn more about molecular basis of inheritance. And let's hope that we keep the session more interactive, and you also freely ask doubts. Okay. Thank you very much, and we will end the session today. Hello, students. Good afternoon. I'm extremely sorry for the delay. Okay, just give me a minute. Okay, I'll get back to you.